Great. Thanks, everybody, and certainly the entire panel for what were really spectacular presentations and discussions about drug development in the early phase. And I think many points were made around the idea that dose derivation and the dose that is derived within early phase trials gets carried forward. It's certainly the fountainhead, and I would argue that much of the data that we get from these early phase trials impacts our older patient population, and probably more specifically our more frail population, even more than the rest of cancer patients who might have otherwise normal performance status and other measures. And so with that, I think we're going to start with the panel and sort of go through and really focus, at least initially, on dose and the idea of dose derivation in early phase trials. And so we'll start with you, Atik, in terms of what we can learn from clinical pharmacology, how we might be able to apply the data of dose exposure relationships, and probably most specifically to this discussion, dose exposure adverse event relationships, and how we might be able to expand that, extend that, and potentially alter labels for reflecting that pharmacology data to improve tolerability of drugs, particularly for patients who are older and more frail.
Thank you, Donald. So my presentation is actually focusing on the dosing issue mostly, as I pointed out in my talk, that there are data that is generated from the non-clinical and early clinical trials where the non-clinical trial, the first thing we take is the toxicology data and a safe dose to start in the first in human trials. But also we have a wealth of knowledge. Just for example, you have the toxicology of the drug in terms of the organs and other systems in our body that gets affected. Even though that are done in healthy animals, we can think about what kind of monitoring we might be needing in the clinical trials as we move forward, and we can include older patients, safely in an early trial, knowing the pharmacology of the drug, knowing a dose that might be safe for general population, and if needed, we can start including older patients. As I've seen that 16% is the incidence rates in most of the larger trials, diseases where larger populations like breast, prostate, colon cancer, non-small cell lung cancer. So if 16% is the patient population, I think there is a patient who would be willing to get into the first in human trial if we can come up with a safe dose. The other thing is that in that dosing, we are just kind of honed on in finding out the MTD or a biological dose to move forward in the general population. And we're not putting our thoughts together how there are information that can be generated very quickly in the early phase trials, such as understanding the clearance, understanding the potential drug interactions. Those are there. You have the non-clinical data to know what enzymes are involved. So you can start to plan early on to think about how safely you can start including older patients in early trials, to start, expanding your inclusion in the subsequent trials safely. And it may be that they would need a different dose, and that's okay. We can take them forward and have a different dose for a certain cohort of patients. We do that for pediatric population in many settings. So it can be applied in that setting too. So my thinking is that the dose is not adequately derived, even for the larger population, let alone the older population. We have data, and I think we don't get to pay attention because of the faster development programs. Neither the industry or the academics research institute nor FDA, nor me, I'm as guilty as I am. We kind of focus on traditional things and move along without thinking that, okay, this is a disease which is prevalent in older patients and how they're included or not included, and what we need to do in early stage to make sure that they can get included in the trials.
Great points. And I think one of the things around that is the idea of therapeutic index. And we think about what we're doing in the world of dose development and early phase trials with the old chemotherapy model, meaning that we've got a dose we know and exposure is too high. it's an drives adverse event. We have a dose and exposure that we might know is too low, but that's an absence of adverse events and perhaps cancer progression. But in these days of newer monoclonal antibodies and other areas where we have a pretty good confidence with on-target drugs, that therapeutic index is much wider, where we're much more worried about the basement of exposure than the ceiling. It seems to me that with those, we can take these criteria for eligibility and put it into an area of really being much more broadly inclusive on drug class, and starting earlier to really open up criteria for that. And so with that, Martine, I'd like to ask you a little bit about what you would change today and how you would change it about eligibility criteria that would specifically move us into a place where we would enroll more older patients with cancer.
So I think we should not be shy of taking on patients with comorbidity or multiple drugs. Okay, the average older person takes 6 drugs, 2 of them interacting with P450. So rather than trying to limit and exclude any P450 targeting medications, what we need to do is, maybe some pharmacokinetics looking for these parameters. And you mentioned that with targeted therapies, it's more finding the minimum effective dose than the maximum tolerated dose. And if you include, a steady state pharmacokinetics with patients who had to reduce their dose, you can find their serum level. You can find if that dose is still neutralizing your target. And I'm aware of a couple of studies where we've learned that. There was an everolimus study that showed that the 5 milligram dose was inhibiting the target. There are some recent retrospective data on venetoclax seem to show that 140 milligrams seems to be inhibiting the target as well as 420. If we could include that in early phase trial, we might be able to broaden our inclusion criteria in a safer way. So that's one of my thoughts, is we have to learn. I totally understand the drive of having as clean a population as we want, but it's less and less representing the actual population we treat. So designing ways of including some comorbidity data and some co-pharmacy data. Mishu Papa is on the call, and Mishu did a study that shows that the drugs that you take with your chemotherapy, if there is a drug interaction, double your risk of side effects, or triple if the interaction is with chemotherapy. So I think this is something we have to pay more attention. It's not that much data because it's usually only baseline data that will matter. Comorbidity doesn't change much over time. So it's just a relatively small piece of data for a huge gain in external validity, I think.
Yeah, no, I think that's an important point, the external validity portion. And with trying to really accelerate drug development, there's a lot of discussion of creating new buckets of patients at the earliest phase of development, whether that bucket is organ dysfunction or others. And so I'll turn now to Jack and Lily specifically to talk about numbers of patients within those buckets and what they can do to help us meaningfully decide dose within those patients, and how would you form those buckets if you were writing the trial or analyzing the trial, meaning let's say we need to create a group of individuals who might be more frail and derive a dose specifically within that group. Would we do it just like a classic phase 1? And what would be the appetite for industry to do that? We'll start with you, Lily.
No, I think a number of points that everyone raised are really important. I do believe there's an opportunity, and there's a real advantage to doing it early because your monitoring is you're on top of things. You've got an intense monitoring, you've got a clinic devoted to phase 1, and this is really the time to do it. I will say I've spent my career doing a lot of adaptive Bayesian designs, and they really give you the advantage of being able to ask specific questions. It's been very easy to add a food effect study early on and add that question, particularly when you have a positive food effect of the drug. Then you really get the excursions. I think there is opportunity to define cohorts that tell you, that look at certain patients with certain types of drugs. The question is, how do you gather the data that you need to communicate potentially to the health authorities to be able to add those cohorts? Because these are multi-site studies, and so everybody wants to make sure you're looking at safety. So that's where I think there's this tension of how fast you can go, with how much you have to look at the prior data and be very confident you can add those other cohorts. I think there's a lot of opportunity. I think that the designs allow you to do that. But Jack, you could probably speak to that even better than I can, although I'm a fan of them.
Thank you, Lily. Yes, from the statistical point of view, we need to use the information more efficiently, right? So we know that there are different patient characteristics and different buckets, as Don said. If we treat them separately, then we don't use that information efficiently. But if we lump them all together, they are not all the same, right? So how can we do it better? So as in my talk, one way to do it is using the Bayesian hierarchical model to borrow information. But not only that, I think we need to think about doing more modeling and adjust by covariates, right? And we need to do this not as a discrete form. We need to do it more a continuous form, more kind of adaptive, like Lily said. That again, I think our clinical trial design, or actually the way we develop clinical trial, is too discrete. Okay, even phase 1, phase 2, phase 3, and trial, they are all different, right? So as I also mentioned briefly in my talk, we need to consider more like a platform kind of design, right? So that can adjust what we learn from the earlier part of design, and we can actually more quickly do some adjustment and either adapt some of the inclusion or exclusion criteria, or in terms of finding the dose, that's more suitable for a certain type of patient. So the key is, again, we need to have a continuously learning kind of system and adjust what we learn in the trial for the subsequent conduct of the trial, rather than kind of very discrete process. So I think we have a lot to learn in that area.
So Jack, you've been working in this for years, and we've read your innovative ideas and the work you've done behind it. Why is it not in practice all across the country? What's happening? Okay,
very good question. I have to say, statistician, in old days, or not even so old days, we tend to live in our own world, okay? So the incentive for statisticians, especially in academic research center, is to publish statistical methodology papers. Okay, so we do have great idea, and they are very nice method. But question is, like Ishwari said, that are they being used, right? I mean, what's the impact? So we need to collaborate more and also change the mindset of statistician. It's not like publish another paper in the first rated statistical journal that matters. It's actually get that implement is that matters, right? Okay, so from that point of view, again, I only have very limited time. I went through very quickly, but we developed software as well to facilitate the implementation for such design. So for example, in the video, you see, you can actually do that yourself. Okay, so just to remind people, the URL is trialdesign.org. These are all free, okay? And thanks for Andy Anderson for the support. Our team that we develop many free available software, and I encourage you to try and let me know what to improve, and together we can really make progress.
Well, Jack, I don't think you're going to put yourself out of a job with allowing that to be fully available to all of us. I think we all need statisticians in our daily lives, particularly in the research world. So I'm going to turn now to Mary Lou and ask you, Mary Lou, one of the things that has been brought forward for the idea of dose derivation is a couple of patient-directed approaches. One would be patients could theoretically choose the dose that they might want to receive. Another one is that a patient might have an intra-patient escalation, that they start at one dose of a drug, and then at some period of time, that patient goes up on the dose if they're tolerating it well. And then we have some analysis at the end of the day where we say these characteristics or this number of patients were able to tolerate this drug based on these covariates. These might be very large studies, but are, as we've talked about, difficult to do. From a patient perspective and a group perspective, how do you see the best way to approach what we're all talking about, which is heterogeneity of dose and tolerability in older and perhaps more frail patients?
That's a hard question, obviously, to answer. I do think the advocacy community is very interested in getting away from maximum tolerated dose because it's going to be too much for some, not enough for others. We're talking about personalized, individualized care. Why can't we bring that to dosage as well? But I also think we need to look at kind of the relationship between the patient and the physician. How do you get them into these trials, right? For me, a little bit, when we get to dosage, we're in the weeds. And we need to back up a little bit, especially when we talk about tolerability, to where the patient and physician are communicating. And I don't think the communication is terribly good when you get to older adults. I'm not sure whether some of it is ageism, or I'm not sure it's some older adults are hard of hearing. And in fact, I know a friend of mine who actually got taken off treatment because he just kept nodding. And people thought he was saying, yes, he wanted to be taken off treatment. No, he hadn't heard a thing that was said. And is it family? Family is a big part of an older person's life. And are they supportive? So I think it's a much bigger issue than just dose. Mm-hmm. I think it's about how you communicate. And I don't think we put much time and effort into that communication and making sure that the patient and family actually understand what's being said to them. We have an awful lot of medical jargon that just gets everyone a little lost.
Mm-hmm. How can we help older patients advocate for themselves and their families advocate for them to their physicians, to their cancer treatment physicians?
Well, I loved what Mary Whitehead had to say about having an older advocate group, having peer-to-peer counseling, so to speak. I think those are ways. I also think we could be doing more of having focus groups and interviews and surveys of older people about, would you be willing to go into this kind of a trial? Do you understand what this word means? So we have messages that we give to older patients that they can understand and accept. So I bet, again, I guess I'm back to communication for me is pretty key.
It makes perfect sense. I loved your comments around the informed consent document that I think we can all appreciate. That's a legal document. It's not really, it doesn't particularly inform, and in all honesty, it's not that much of a consent when it comes to legal ramifications. So I appreciate your comments on that. I also appreciate your comments on having housing and for those early phase trials, where we do ask a lot of patients to be with us for 12 hours, multiple days during a month, come from long distances. Your own story about traveling to Minnesota from Chicago, I think is very, rings true with a lot of us here in terms of how we see our patients come into trials. So let's pivot now to Michael. And Michael, I want you to sort of hone in on your idea and what you mentioned about geriatric use and labels. If you could wave your magic wand there in Northern Virginia and change a label that would be more inclusive for older adult and frail and geriatric dosing, what would you do and how would you change it?
It's a good question, and I hate to sort of derail the specificity of our workshop, but this is, a global issue. So let me try to put it into a little bit of context as I answer your specific question. As a member of the clinical pharmacology community, you share my frustration that's now more than a decade long in terms of how this rapid pace of developing drugs, for very good reason, right? We want to get new effective agents to patients in need as quickly as possible. But that has been done through this method of developing drugs with empiric methods. So we give a dose, and then we aim to determine a maximum tolerated dose, and somewhere in the middle, we figure out what is the dose we're going to move forward in phase 2 clinical trials. And then effectively it's left to a basic guidance and physician experience and sometimes good communication with patients to modify the dosing from there. In other fields of medicine, where there's a less sense of urgency to bring a new product forward and a much higher bar for safety and also sometimes efficacy, a lot more time is invested in understanding how patients vary in their metabolism of the drug at a certain dose and how that affects the side effect profile and the efficacy. A deep evaluation, getting into the weeds for you, Mary Lou, but I think this is actually quite important to understand, is exposure response. Mm-hmm. And so I think the drug development and oncology community at our side of the interface with patients that need to invest more time and resources interpreting those data and maximizing their value as we bring forward, I think, that are a range of doses out of phase 2 into phase 3. So I think that's issue one, is just that bread and butter clinical pharmacology. Mm-hmm. I think the second issue that I've experienced personally with the benefit of interactions with some of the brilliant people, who've organized this meeting, like Dr. Mohiel and Dr. Musk, is this is interdisciplinary. And it's a huge problem, and we've had many members of both panels identify a host of different issues and obstacles. And this characterizes fundamentally problems with interdisciplinary research and innovation. As Dr. Mohiel said, we're all in these silos, and so we can keep being productive as academicians or drug developers if we stay focused and don't spend too much time working with others to advance a bigger goal. So I think there's a real shortage of investment in the innovation. And I think there are many policy solutions for that. So I'll save the rest of my comments there. But ultimately, the answer to your question, Dr. Harvey, is we could better adopt in early development the methodology of other fields of medicine. And I think, as some folks have been alluding to, in the post-marketing setting, a greater investment in collecting important data that we didn't get in the early development.
Mm-hmm.
So the solutions proposed here really warrant a change in the phase 1 clinical trial design that happens months to years before a site would see the protocol, right? And so let's go to Lily. We'd love to hear your thoughts on how to integrate these changes at the trial design level before, frankly, many of the people on this call may come across the study.
I actually think you can think about the questions to, one, you know a lot about preclinical behavior. You know a lot about things you're worried about in a molecule. You have in your head the questions you might want to ask. It is, and how do you adapt? And I think it's doable because I've been lucky to do mostly adaptive designs my entire time in industry. I came from academia, and then I went in and I didn't know better. So I said, how do you get the most information you can? And so I've done a lot of hierarchical designs, looking at different doses of different diseases. And I also think there's the potential, and I think it's right in front of us, but you have to have the statistical expertise to say, okay, I am going to change an inclusion criteria once I know I have a data set and have evaluated it. But you're responsible in the industry side for pulling in the data from multiple sites. So you really have to decide what's enough data and be flexible to really analyze when you need more data. And so you do have to be able to build models that take your preclinical data and then the dose exposure data you're getting in clinic, as well as the toxicity. And so you have a lot of tools to build all that, but it takes a lot. I am never, I think I did 3+3 dose escalation in my career. So, and I have to say, I haven't done many because I like these ideas that you ask questions, and then you have to say, when is there enough data? And you need a statistical partner for this as well. So I think we're there. The question is, we're not there at every place or every time. And then there is that desire to move fast. And I think you can move fast, but you also, if you think about the questions early and build it into the study, because every time you amend to add different things, it is time consuming and adds additional cost and everything. So that's my thinking on it. I think we're not as far as people think, but the expertise is not everywhere.
Jack, your thoughts?
Is that?
Yeah.
Yes. May I just add one more point? Again, like in traditional phase 1 trial, we want to identify the maximum tolerated dose, right? And there has been said that maybe we need to identify minimal effective dose or even optimal biological dose. But when we talk about this term, it's still just one dose for everybody, right? So think more about this personalized dose, like Mary-Lo also mentioned that, right? I mean, everyone is different. And how do we do more modeling, more kind of adjust by covariates, and including preclinical data, as Epic talk about. But this is hard, okay? But we have the infrastructure. That's why we haven't done this, okay? But I think we are at the verge that we have the data and we have the computing power and we have the method, okay? So like Lily said that, I mean, she has been doing that for a long time, but I think right now we are at the verge that we can do something really kind of revolutionize things, more novel, more revolutionary things. But one thing very important from statistical point of view is validation, okay? So we can definitely do a lot of exploration and find out, build up some risk model or the building pharmacology, pharmacodynamics, etc. Okay, this is all good. Okay, but then we definitely need to have some kind of validation. So I think people talk about this learning healthcare system. Okay, I think we need to also think about that in the drug development, and so that we can really revolutionize the way we run phase 1 trials.
If I may make a comment, the one point we have not yet addressed is the issue of body composition. When we zero down on a healthy group of patients, we have a fairly homogeneous body composition in these patients. But then as we age, two things can happen. You can either become very fat, or you can become sarcopenic, or you can be both. And this definitely has a high influence on your volume of distribution, and it's poorly explored in early studies. And nowadays you can do body composition on a CAT scan. So I think that should also be integrated in our drug development.
Yeah, no, certainly tolerability by measures of L3 cuts and albumin measures and others can certainly help us to potentially predict a lot of these items. So, other comments? I think we're kind of nearing the end of our time. Is that right?
Yes, our time is up for today. We could continue this for hours, but we'll hand it back to Monica.
Yeah, I think we want to thank again, everybody. I want to thank my colleague Ishwarya and all of our panelists on this session, and certainly as we try to get to solutions for how we might improve our information around older adults.